So in this section, we're going to talk in more detail about some of those genes that are responsible for taking the pluripotent cells and turning them into tissue-specific cells like muscles and bones and brains. Now, most of this work was done in the fruit fly Drosophila melanogaster, but as it's a model organism, and as much of what's going on here is conserved across all animals, it works great. You could have chosen just about any organism to do it, and many of these things would have also been discovered. So, although we're going to be looking at discoveries made initially in the fruit fly, these have been extended to all animals. So, there are about 200 or 300 genes that fall into this class of what we would call homeotic or developmental genes. Now, one very important subcategory of these homeotic genes are the Hox genes. And those are the only ones we're going to talk about in any detail for this class. So, Hox genes, which is a short for homeobox genes. Now, these are all transcription factors. Now, we mentioned this before, but when I say transcription factor, in your head you should be thinking of a gene that when it gets transcribed into an mRNA and then finally translated into a protein, the job of that protein is to bind to DNA and then help to regulate, that is, to turn on and turn off the expression of target genes. Okay, and so all of these homeotic genes, especially these Hox genes, are transcription factors. Okay, so just keep that in mind. And very early on, a small subcategory of these homeotic genes was found that were critically important for the development of the fruit fly, and then later that was extended to all animals. So these genes are found in a order that is called collinear. So I'm going to skip ahead since that's where I'm at right now. But collinearity means that the first gene controls the expression of the anterior portion of the head. The gene right next to it controls the middle portion of the head, the gene right next to that, the posterior portion of the head, and so on. And this is very unusual. Genes usually can be found anywhere in the genome, and as long as they're able to be turned on and used when they needed to be, their location relative to other genes, where they're found like next to gene A or gene B, usually doesn't matter. But for the Hox genes, it's critically important. And in nearly all species, this collinearity is maintained, that these genes are all found right next to each other in the same order in which they control the development of the animal. Okay, so anterior ones are found next to the ones that are next most anterior, all the way down to the posterior. So that's collinearity, know what it means. Now, the reason for this collinearity is that there is critical regulation of these as a group. And so mutations that move them to different parts of the genome, which we learned about, right? That's called synteny. And we learned about in the genome section that happens quite frequently. And so it's not that these mutations haven't occurred. It's just that when a mutation occurs that moves a Hox gene to a new location, it's not a good thing because they need to be all together so they can be regulated and used together. Okay, so that's collinearity, and you should know what it is and the reason for it. Let's go back to number 2. When we find mutants of these critically important Hox genes, we almost always have a homeotic transformation, like antenna growing in the wrong place. And in fact, many of these genes are named after the homeotic mutation. So here's a Hox gene called antennapedia. When we mutate that, we get antenna growing legs growing where the antenna should be. Some are called deformed, just because that one was a little less of a clear one. There's one called UBX, or ultra bithorax. When we mutate this gene, we get flies that have wings in the middle section of the thorax and in the hind section of the thorax, which most flies don't have wings there. So these mutations, this is an antennapedia mutant, this is an antennapedia and proboscipedia mutant, a double mutant. And so you can see these changes. This one on the left is just a wild-type fruit fly. And so a tiny change in these critically important gene results in a major shift. And again, these are like any other mutation. Most of the time they're bad. They won't be kept by natural selection. But every once in a while, one of these homeotic mutations results in a very advantageous phenotype. The fourth characteristic of these Hox genes is that they are found in all animals. Now, I want to make it clear because I find very commonly that when we say animals, biologists, even biology majors, will have something in their head that's confused. So animals is the group Metazoa. Okay? So humans are animals. All mammals are animals. Arthropods are animals. all of the mollusks are animals, right? So an animal is any organism that is, one, multicellular, and two, has nerves and muscles that allow it to move around. And you could add some other things onto that, but those two are sufficient to define animals. A multicellular organism with nerves and muscles so it can move around. The metazoa is the name of the scientific name of the group. And so these Hox genes, although some have a lot and some have relatively few, is still a critical feature of all of the animals. So basically, these Hox genes are the major diversification force at the genetic level that helps create and generate diversity in animals. Okay? Now, here is a review of what we've already outlined about these genes. So the Hox genes, other genes too, but the Hox genes are transcription factors. They work sometimes singly or in groups like we have here to bind to regulatory sites near a gene. So here's where the gene is. They bind to it. That recruits the RNA polymerase to come in and transcribe this gene over and over again. If those guys aren't there, that gene doesn't get transcribed. So these guys basically work as master control switches, as mechanisms that will turn on the expression of the gene. When that gene is expressed, then it can have its impact on the developing organism. So, and this will be critical for our review and understanding of that initial hypothesis we talked about in the last discussion, right? So remember, the initial hypothesis is that new genes or different genes in the fruit fly make a fruit fly grow into what it's going to be, whereas in a butterfly we have a completely different set of genes. Okay, so we're going to set this up and we'll revisit at the very end of 3.3D. So realize that, and this is just one way a major change can occur. So here we have a transcription factor, the purple one here, with a specific binding site. And we have these two genes that need to bind to help create a wing like this, two genes here that make a black abdomen. We end up with a fruit fly with clear wings and a black abdomen. Okay, so this gene has two jobs. This is a pleiotropic gene, right? It doesn't just do one thing. It controls pigmentation of the abdomen and also pigmentation or the lack thereof in the wings. So if we add a third binding site, so we have a mutation in a different species, or maybe originally it was part of this species, but a mutation occurs there, so now we get a wing spot. Now initially this just might be variability in the population where some of the individuals of the species have clear wings and some have wings with a wing spot. But eventually, if that was different enough, or if it created enough behavioral differences, we might get further separation, and we end up with a different species of fly. Here's a third one where we don't have that binding site here, but we end up with a loss of a binding site in this portion of the regulatory elements, and that results in a non-pigment abdomen and a morphologically subpopulation, again, maybe even eventually a different species. Now this is key, and we'll come back in when we revise and revisit our initial hypothesis. Notice that we had dramatic changes in these organisms, maybe even enough to define separate subspecies or completely separate species. But notice the gene itself didn't change. All we did was mutate and change the ability of transcription factors to bind to their regulatory elements and have an impact or have a controlling influence on the gene. The gene itself did not change. So we have three species now here with the exact same gene, or potentially the exact same gene, but with different regulatory binding sites that allow it to use that gene differently. And this was the first clue that this initial hypothesis maybe wasn't the most important. So the initial hypothesis was different genes making different organisms. We're going to revise that at the end. And this is the first clue that maybe genes could stay the same or largely the same, but yet have a dramatic impact on different populations based on how they are turned on and how they are regulated.